What's up, folks? So today I want to talk a little bit about the cytoplasm, and I'm sure you've heard this term used before in biology, but it's basically just there are components that are used in a cell to take do certain things. You know, I so I guess I'll first talk about like what the components are. So the first one I want to talk about is called an actin filament. These are made of actin, which is a protein. You might have seen them called microfilaments. Basically, these are just little. They're made of F actin and G actin. They're pretty short lived. They're made to do a process, maybe move the cell or this thing called cytoplasmic streaming in plants, and they don't last that long, right? So I'm just gonna write an A next to it. The next one I want to talk about is intermediate filaments, and these are basically the medium size ones out of there are three. So the third one is microtubules, right? And these are the biggest ones, right? So if you maybe have a little bit of trouble remembering, I use the mnemonic AIM. So that if I forget one of the three, and the good part about this is like small, medium, large in size relative to one another. So they're it's not that bad to remember. Um, Now, you know, before I move into talking about the other two, I think I want to focus on actin a little bit. So these are roughly 7 nanometers in diameter. Um, so they use a motor protein called myosin, right? And let me say D is 7 protein. I mean, D is 7 nanometers. Myosin is motor protein. And basically, um, actin is used for, so there are a few different things, right? One of the main points of actin is to resist tension. So you're going to find that all three of these either do something in resisting tension or resisting compression, right? So tension is like pulling, right? So if you pull on two ends of a rope, right, you're putting tension in the rope, and basically actin helps to resist types of pulling, right? Other things that it does, basically, if a white blood cell is chasing a a pathogen, right, you're gonna find that the white blood cell, right, that pathogen is gonna release. I'll just do this, right. It's gonna release little these things we call ligands, and they're going to be receptor proteins. You're just going to see it drawn as a Y. I don't actually know what it looks like, but you're going to see those receptor proteins are going to maybe get like pulled into the, well, they're going to end up like getting into the receptor protein, and then that receptor protein is going to lead. There's this thing called a signal transduction pathway. It's going to lead to the creation of second messengers, right? And then those sort of trigger the white blood cell to start building microfilaments in the direction that they were, that the second messengers were produced. So those microfilaments are going to cause the red blood cell to start moving. They're going to push against that red blood cell. I mean, I said red blood cell. This is a white blood cell. I'm sorry. White blood cell. So like they're going to push that end of the white blood cell, right? And it's going to start moving toward that pathogen, maybe to get rid of it, right? So I'm just going to say white blood cell movement, right? What else do they do? I'll just go ahead and maybe if you want to just copy that down as I'm talking. I don't know. So I'm sure you've heard of this thing called cytokinesis. They're used during that as well. Now, cytokinesis is basically there's a point in time when cells have to basically continue to reproduce or they'll die. I know that sounds sort of weird, but if a cell gets too big, basically it becomes very much less efficient. It can't, distribute the nutrients that it needs and everything as efficiently. So that definitely is that's definitely something that the cell needs to do to survive. So cytokinesis, you're just gonna have maybe So like maybe like if this is the cell getting ready to split, right? You're gonna have maybe actin filaments that are on the inside of the cell that continue to pull it closer, right? And pull it down, 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 down until like that cell splits, right? So sometimes they say it's like a pinching of the cell, so it splits the cell in two, but these are actually on the inside of the cell, right? So that's cytokinesis. And I want to emphasize that this cytokinesis is in animal cells rather than plant cells. Microtubules are actually used in plant cells. Oops. Another thing that they do, cytoplasmic streaming. Now, this is actually something done in plant cells. So let's say we have a plant cell here, and plant cells are actually depicted as like square. So you're gonna have like the nucleus toward the center, right? But you're gonna have like those actin filaments sort of pull those nutrients around the cell to be distributed around that cell. So some microfilaments also help to do that, and they're also used for muscle contraction. Muscle contraction. Yeah. So do I want to just make everything into one video? Yeah, let's go ahead and just talk about intermediate filaments. So intermediate filaments are another type of element, right? I'll just write it as IF. Now these, let me just not be lazy. Let me just write the whole thing. So intermediate filaments are more used for structure. Intermediate filaments. Now these are about 10 nanometers in diameter. Well, 10 to 12 actually. So these are basically like medium sized out of all three elements, and they actually don't have a motor protein, right? And that's because they don't really need to move around a whole lot, right? Their main thing is to make a structure stay, right? That's really the point of intermediate filaments. And the way I sort of remember intermediate filaments, and I don't know if this directly relates, but your hair. So actually they are, and I don't think I said this. I probably said it, but I don't think I really emphasized this. But these are made of actin filaments. And I think I should point out, and I don't want you to get this confused. Myosin is not part of an actin filament. Myosin is a totally separate protein, right? And that protein works in conjunction with actin, right? And myosin sort of looks like a golf club, and it might look something like right, and then it moves back and forth, right? So like that cell, not cell, that protein is gonna move back and forth, right? And it does this thing that's called a power stroke to allow it to like it'll grab the actin and move it, right? So it's grabbing it. So that's I just wanted to point that out. Now these are made of keratin. Well, I should say keratins because there are different types, right? Now, keratins, like that's why I said that's what was taking me to the hair and the fingernails thing, right? So your hair and your fingernails, I've always heard though they're made of keratin. They're made of keratin. So that's actually what helps me to remember what an intermediate filament is, right? These are long lived, right? So if I don't cut my hair, it's just gonna stay there. Yeah, hair falls off after some time, like their hair cycles and stuff, but like hair for the most part is gonna stay on your head, unless maybe there's something that like isn't how it's supposed to be. That hair is mainly gonna stay on your head. And the same thing with fingernails. Fingernails aren't just gonna disappear after a while. So that's how I remember intermediate filaments. And these, like I said, are mainly, so actually first I should say they resist tension as well. And these are mainly used for structure and support. And what do I mean by that, right? So, most times we have, oh, wow, I don't know how that happened, but we have a nucleus, right? And, the nucleus has a double layer, right? Double plasma membrane. And, so you're going to have intermediate filaments on the outside of the nucleus, and they're so they're gonna, whatever. Those are gonna, not stretch out, but they're gonna attach to that part of the plasma membrane, and you also have some on the inside, right? Sorry, I'm just trying to draw them, right? So we have intermediate filaments on the inside as well as the outside, whatever. And those are gonna, support one. They help to keep the nucleus in a so I should actually say these right here are keratin proteins, right? These are the intermediate filaments, right? Also made of keratin, right? But also made of those keratin proteins. So basically, these keratin proteins help sort of keep that circular shape, or of the nucleus, right? And they also attach the chromosomes, because the chromosomes rest in the nucleus, so they attach them to where they need to be, right? Now this right here is the nuclear cage, right? This is what sort of holds the nucleus in place, right? So these keep that nucleus circular, while these actually hold that nucleus, and you don't want the nucleus just, I don't know, I guess bouncing around the cell, right? That's not going to be very good. So that's the main role of intermediate filaments. And lastly, you have microtubules. And these are the biggest ones with the diameter of 25 nanometers. So those are substantially bigger in diameter than your microfilaments or your intermediate filaments. And the protein of these, this is sort of easy to remember. Tubulin is protein subunit. And so these actually do have a motor protein. These are keratin. Well, I said keratin. These are kinesin. And I should also say dynein is the motor protein of microtubules. And what do these? Well, okay, first I should say these are the ones that actually resist compression, right? So these, the way I remember it, I don't know how effective this is of memorization, but I just think about it. If I try to push on a pipe, right, and my engineering brain just goes to like, we have this, there are certain shapes that are more resistant to, like compression, right? So if I have like a pipe, right, it's gonna be very hard for me to push that pipe and like maybe like compress it, right? So if I put a downward force here, it's gonna be sort of hard for me to force that pipe down. So that's the way I think about it, and so they resist compression. They are mostly short-lived, but they can be long-lived. And microtubules, what do? They sort of serve as a, I'll say, pathway for different things. Now, what do I mean by that? So, one of the things microtubules do, during mitosis, they move like, those chromosomes, right, to where they need to be. And, there's this thing called the spindle apparatus, and the motor protein dynein is involved, right? And I'm sorry, is that actually, what? I'm not too sure about that, actually. Let me not say that. However, there is a motor protein involved. And, I'm just not too sure if it's dynein or kinesin. I want to say it's dynein. Okay, I'm not too sure. But basically, those move the chromosomes, maybe pull them apart to where they need to be. You're also going to find, now cilia and flagella, those definitely use dynein. And, they also use, they use dynein. So, let's say there's this thing called an axoneme. And that's this structure here. And they have a 9 times 2 plus 2 structure. And what do I mean by that? So they have 9 groups. These are attached, right? 3, 4, 5, 6, 7, 8, 9, right? And then they have 2 singular microtubules in the center, right? And these have a name. These are called doublets. Sorry if this video is running sort of long. I maybe should have split it up. But basically you have let me just try and speed it up for y'all. But like you basically have these little connectors, and you have dynein around them, you know, and that dynein helps to basically like, you know, pull, push, you know, like back and forth, and it creates this like wiggle movement. I'm sure you've seen like maybe videos of a sperm cell moving. That's it's dynein that causes that movement, you know. And this 9 times 2 plus 2, there are 9 pairs of these doublets, right? I should say there are 9 doublets, right? Plus 2 single microtubules in the center, right? So there's also there's also cytokinesis in plant cells. And I don't even remember how that works, but basically, you know, those microtubules again, they're gonna be transporting something throughout the plant cells, okay? And you know, these also you're gonna see, you know, they have these things called centrosomes. MTOC just stands for microtubule organization centers. These are in animal cells, and basically these I think they said they don't know what they are actually used for. I can't remember too much, but basically centrioles make up a centrosome, right? So I'm sure you've seen maybe some things of like this, and I don't think they know exactly what they do, but that's basically a breakdown, right, of just basically the 3 of these you know, cytoskeletal elements. Really, they're pretty useful, you know. And you know, the first time I took biology, maybe like when I was in high school, I didn't exactly know what was going on here, right? I didn't understand how these were related, and maybe I think I just aimlessly just tried to, oh, okay, microtubules do this, you know. But basically, these are all elements that help the cell to function, right? So you have, I think, billions of cells in our body, right? And each cell, well, I mean, I think it's depending on how specialized the cell are. It's gonna be for different things. Like this is my like white blood cell movement and everything. But like, basically, they're used for different things, you know. And it's very beneficial to the cell to be able to have these elements, you know, do what they need to do for the cell, you know. So it's not too bad, honestly, once you break it down and see what they do. And you know, maybe once you link these to other things, you know, like phagocytosis, you know, you can see exactly how the microfilament like causes this movement and causes it to engulf the bacteria, and how that works with other parts of the cell to maybe, you know, process the bacteria, get nutrients from that bacteria and everything. So I'm gonna go ahead and end the video here. Thank you so much for watching. If anything was unclear, please go ahead and ask in the comment section. If you like this video, please go ahead and give me a thumbs up. I guess if you didn't like it, feel free to give it a thumbs down. It'll just give me feedback, you know. But thank you so much. Please feel free to subscribe to my page. I would really appreciate it. I also have other videos, physics, engineering, things like that. So thank you so much, and let's keep learning, okay? Bye-bye.